Hello, and thank you for visiting my poster. My name is Aarthie, and I'm a student at the University of Ottawa. I'm excited to share with you my recent work on novel real-time computational assessment of protein differential expression during mass spectrometry experiment for the optimization of data acquisition. Mass spectrometry is an effective method used for analyzing protein samples. When coupled with tandem mass tags, TMT, it is a powerful tool for quantifying proteins across multiple conditions within one single experiment. However, one significant challenge remains that this type of data acquisition method is biased towards the redundant data collection of high abundance proteins, and this redundancy leaves many low abundance proteins unrepresented, limiting the comprehensiveness of our analysis and experiments. So some related work has focused on analyzing good quality spectra. However, this favors highly abundant proteins and leaves thousands of proteins without enough quantification information to assess their differential expression. So my goal was to design a computational exclusion method that assesses protein differential expression confidence in real time during a TMT mass spectrometry experiment to limit the redundant data collection from high abundance proteins. So how do we do this? Once an MS1 spectra appears in an experiment, we can check to see if it's on an exclusion list. If it is not on the exclusion list, it undergoes fragmentation, protein identification, and quantification in real time. Now, once two or more spectra are collected for the protein, we can now calculate the confidence interval for the quantification values using TMT intensities. Based on our confidence interval, if it falls into one of these three categories, it will be deemed as confidently quantified, and it will be added to an exclusion list. This will prevent any further collection on this protein within the experiment. However, if it is not, we continue collection on this protein. So I tested this method on two different data sets. The first is a HEC293 cell line using a TMT11 plex, which compared a control and cells treated with TORN1 for differential expression. Next was a TMT6 plex mouse intestinal epithelial wild-type cell dataset, comparing a control with cells treated with HDAC1 and HDAC2 deletions to test for differential expression. Here is an example of how our algorithm will work. Here we look at our protein from the mouse IUC dataset. In red, we have our log2 average fold change of the protein, and in green, we have the calculated confidence interval for each spectra collected for this protein. Once the confidence interval meets the requirements that the protein can be excluded, this will prevent any further redundant data collection on this protein where we see the quantification values do not change that much. This will ultimately save many resources within a mass spec experiment. First, I wanted to test to see what the potential savings this algorithm could provide. So I used the previously identified protein information for exclusion. I found that we could save up to 37% of MS2 spectra, which leads to significant resources saved. Next, I wanted to see how this exclusion would affect our quantification values. So here, I compared the fold changes between our excluded and our full data sets with the proteins in red representing the excluded proteins. Positively, we were able to see high correlation scores between the two, therefore emphasizing there is very little change in our final results while only using a fraction of our data. The next step was to simulate the real application of this algorithm with the exclusions now based on retention time predictions and mass to charge values, in which we save about 7 to 9% of our resources and maintain 98% of our proteins quantified with high correlation scores above 0.98. I then tested this with different tolerance parameters for the exclusion, and with the most relaxed parameters, we were able to exclude up to 8 to 12% of spectra while still maintaining high correlation scores between the quantification values of our full and excluded datasets. So from this project, we can see that the exclusion of peptides from fragmentation for confidently quantified proteins can save up to 35% of resources which could be reallocated for the quantification of other proteins. With retention time predictions, however, this could hinder the exclusion procedures, but still allows up to 10% of resources to be saved. Overall, giving us extremely great results for resource saving and maximizing our potential on analysis for these mass spec experiments. Finally, I would like to thank the members of all my labs, our collaborators at Bruker, and my funding resources for making this research possible. Thank you.